her screen first. Okay. Okay. Hi, everybody. Welcome. I hope you're all doing well. I hope everybody's stuff is going well, kind of getting back to normal as much as possible. Today we have one speaker or one person that's presenting. Her name's Ashley Stevens. Ashley is a PhD candidate at the University of Kentucky, and we actually started graduate school together. So we've been at UK together for the last 3 years. And Ashley is in Dr. Tianyan Gao's lab here and is I think I already said, a PhD candidate. And today she's going to be talking about PTPRF and oncogenic phosphatase in colon cancer. So Ashley, you can take it away.
Okay, thanks, Caroline. Hello, everybody. My name is Ashley. I'm a fourth year student at the University of Kentucky in the Molecular and Cellular Biochemistry department. And today I'm going to be telling you about PTPRF and how its suppression is tied to Wnt signaling in colorectal cancer. No subtitles. Laser pointer. All right, so the PTP protein tyrosine phosphatase family is divided into 4 different classes based on their catalytic mechanism. My phosphatase that I work on is in the class 1 family. Class 1 can be further divided into classical or dual specificity. Classical phosphatases are tyrosine specific and can be further divided into receptor or non-receptor types. I will be focusing on a receptor type PTP. And so looking at the receptor PTV family, there are 8 different subclasses, and those can be divided based on their extracellular domains with various like cell adhesion binding domains. And so I focus on the receptor 2A family, which includes PTPRF, S, and D. Looking at class 2A more specifically, we see that they have IgG, extracellular domains, as well as fibronectin type 3. There are 2 intracellular phosphatase domains. The D1 domain is active and the D2 domain is inactive. Just generally, PTPRF is the most well-studied of the 3 for its role in axon guidance in Drosophila studies. Generally, PTPRD is a well-characterized tumor suppressor in various cancers, and PTPRS is also involved in nervous system development and is also characterized as a tumor suppressor. And all 3 of them share over 84% identity in their phosphatase domains, but the most variability comes in their extracellular domains, sharing 64%. So looking at PTPRF, again, the extracellular domain is characterized by those IgG and fibronectin domains. The intracellular phosphatase domain shown here in the crystal structure has D1 domain active, showing the catalytic cysteine residues here, and the D2 inactive domain. PTPRF is cleaved post-translationally, and they associate non-covalently at the membrane. And this step is important in the Western blots I'll be showing. So looking at the tissue distribution of PTPRF, we see that it's highest in the colon and small intestine, which is important for my studies, as well as in the fat and skin tissues. And a main difference between the class 2A is that F, S, and D do share various tissue expression differences. So what's known about PTPRF? Generally, its main role is in axon guidance, and that has to do with its extracellular domain functions. A recent study came out showing a mouse model with a deletion of RF and RS, and that showed a craniofacial development defects. Its involvement in cell polarity has been characterized, and there's only been one true substrate identified that it dephosphorylates, and that was identified in 2012 as being Epherin alpha 2. There are other proteins that it does interact with, including the insulin receptor, syndecan, and PPFIA1, or also known as liprin alpha-1. These proteins either interact with the extracellular domain or the inactive phosphatase domain, but none have been shown to be true substrates. And then PTPRF's role in cancer has been varying based on cancer types. And so it has shown tumor suppressor properties in liver, prostate, breast, and gastric cancers, but has shown to be oncogenic in breast and thyroid cancers. And so we wanted to investigate its role in colon cancer more specifically. So as a lifelong Kentuckian, born and raised here, this is not a stat I'm proud of, but one that needs to be highlighted. So Kentucky is shown here. We rank the highest in colorectal cancer incidence rate, and that's shown here, the top 5 states. Kentucky is the highest in colon cancer rates. And so obviously we need to study this more and better understand it. So colorectal cancer is defined as the inappropriate proliferation of the intestinal epithelial cells. And overall, it is the second leading cause of cancer-related deaths among men and women. And so Wnt signaling is defined as a adenoma to carcinoma sequence where over the course of cancer development, more mutations in various signaling pathways are acquired. And so the first hit is usually a Wnt signaling pathway mutation, which I will be focusing on. So looking at the Wnt signaling pathway, it can be defined in an off and on state. So without Wnt ligand present, the co-receptor LRP6 and the Wnt receptor frizzled are inactive. This leaves the destruction complex in the cytoplasm free to phosphorylate beta-catenin, targeting it for degradation. This keeps target gene transcription turned off. With Wnt ligand present, binding to its co-receptor LRP6 and frizzled, this tethers the destruction complex to the membrane, allowing beta-catenin's accumulation, translocation to the nucleus, and Wnt target gene transcription. And these genes are important in cell proliferation, stem cell fate, etc. So knowing that PTPRF's role in cancer was varying and we wanted to understand that more, we first started to look at PTPRF's gene expression in colorectal cancer. And so looking at PTPRF here, we see that it has some mRNA upregulation. Using a gene set enrichment analysis, we also showed that colorectal cancer, that PTPRF positively correlated with it, as well as Wnt signaling. And so with this initial data, we wanted to further characterize PTPRF's role in colorectal cancer cell growth. And so this diagram of PTPRF is to better help explain the 3 bands on the Western blot. And so we have the full-length protein, the extracellular domain, and the P domain shown here. And so using a lentiviral mediated knockdown of PTPRF, using 2 different sequences targeting, we see that the knockdown of PTPRF was achieved. Using these knockdown cells to do a cell proliferation assay, we see that in both HCT116 and PT130, 2 colorectal cancer cell lines, the knockdown of RF decreased that cell growth. This cell growth decrease was further exhibited when those cells were grown in 3D, plating the cells in matrigel. So continuing with the cell growth, we next moved to mouse tumor organoids, which we commonly use in the lab. And these were taken from APC-KRAS, colon cancer mouse models. And so we also did PTPRF knockdown using lentivirus knockdown in culture. And we see that RF knockdown changes the phenotype of these organoids. And so when we plate those for colony formation, we see that fewer colonies are formed when PTPRF is silenced. Examining those organoids, we see that they have a more branched phenotype as well. Using those same PTPRF knockdown organoids, we used click-it EDU staining to assess the proliferation. And so at day 3 of growth, we see that RF knockdown organoids have significantly less EDU staining, and that's quantified here. So not only does PTPRF slow the growth of colon cancer cells, but also proliferation in the tumor organoids. Examining various Wnt target genes using qPCR, we see that those tumor organoids, here's a validation of PTPRF knockdown, and whenever we see that LGR5 and cyclin D1, 2 Wnt target genes are also reduced. And so this is also replicated in the HCT116 colorectal cancer cell line, showing that reduction in Wnt target gene expression. So looking more closely at how PTPRF affects Wnt signaling, we use the commonly used reporter assay in the Wnt field, the Wnt luciferase or top flash assay. And so this allows for Wnt activation to be read out as luminescence. And so using control and PTPRF knockdown cells with a various time treatment of Wnt for 4, 6, or 24 hours, we see that PTPRF knockdown cannot achieve the same level of Wnt activation compared to control. This knockdown was validated here, and then we also see that active beta-catenin levels are decreased in the PTPRF knockdown cells as well. Complementing this with an overexpression of PTPRF in the top flash assay, we see that we get an increase in Wnt activation as well as an increase in active beta-catenin, suggesting that PTPRF is helping to activate Wnt signaling. So next, we wanted to ask where specifically in the Wnt pathway PTPRF was acting. And so the Wnt pathway can be thought of as in either upstream or more downstream, either at the membrane level or the post-destruction complex. And so we used a GSK3 inhibitor, which is part of the destruction complex. This inactivates GSK3, therefore activating Wnt downstream. And so using qPCR, we see that the RF knockdown decreases Wnt target genes in both HCT and PT130 cell lines. Addition of that GSK3 inhibitor activates Wnt, but also activates the RF knockdown cells to the same degree, therefore suggesting that PTPRF functions upstream of the destruction complex because it was able to be rescued by that inhibitor, which makes sense because RF is a membrane PTPRF, or a membrane PTP. So next, we wanted to identify more specifically what in the Wnt pathway PTPRF was affecting. And so a common marker for Wnt activation is the phosphorylation of serine threonine residues on LRP6. And so using the same knockdown of PTPRF, we see that phospho-LRP6 is reduced, and that is quantified here. And so this decrease in phospho-LRP6 is suggesting a decrease in Wnt signaling, which matches our top flash assay. So next, we were looking at PTPRF's interaction with LRP6 more specifically. So these are single color expression controls of RFP tagged PTPRF and of GFP tagged LRP6. We see both are membrane bound as well as in some vesicles here. And so when those are co-expressed together, we see that their interaction is at both the membrane as well as in intracellular vesicles. And so this interaction with LRP6 in these vesicles was interesting and therefore led us to look at PTPRF's role with clathrin endocytosis. So LRP6 is known to be endocytosed, and so we wanted to know if PTPRF required this as well. So using the top flash assay again, we added the pit stop 2 clathrin endocytosis inhibitor. And so overexpressing PTPRF again led to that activation of Wnt signaling. However, addition of that clathrin inhibitor reduced RF's activation. This was repeated in HCT116 colon cancer cell line. Overexpressing PTPRF, we get Wnt activation, and the pit stop clathrin inhibitor inhibited this activation, suggesting that PTPRF's activation of Wnt signaling is clathrin dependent. And so finally, we moved into a in vivo tumor xenograft model to assess PTPRF's role on colon cancer growth. And so using PTPRF knockdown cells, over 6 weeks allowed the tumors to grow and saw that they grew significantly less. This was also quantitated by tumor weight, showing that they were significantly smaller. We then analyzed these tumors and showed that the knockdown of PTPRF was here, as well as the decrease in phospho-LRP6 expression. This is quantified here as PTPRF levels, as well as a decrease in phospho-LRP6. So this was replicated in tumors as well as in cells. We then used qPCR to look at Wnt target gene expression of the tumors, and we see that there are Wnt target gene decreases. And so this is suggesting that PTPRF does have a function in Wnt signaling, acting as an oncogenic phosphatase in colorectal cancer. And so in summary, I have shown you that the knockdown of PTPRF did decrease colorectal cancer cell growth, that had a decrease in Wnt signaling. PTPRF acted upstream of the destruction complex. PTPRF interacted with LRP6, and that RF's activation of Wnt required clathrin endocytosis. And then finally, that PTPRF knockdown inhibited tumor xenograft growth, and that those tumors showed decreased Wnt signaling as well. So with that, I'd like to thank my mentor, Dr. Tianyan Gao. She's amazing. Tong helped me on this project. He was a surgery fellow. Xiaopeng, our postdoc, Sumati, and Stephanie. And I'll take questions now. Thanks, guys.
I always hate that part. We all want to clap. Yeah, if anybody has questions, we'll open it up, or if you want to ask questions in the chat, Jesse and I can screen those too.
I have a question.
Oh, sorry, go ahead.
Do any of these effects depend upon the activity of LAR?
So I am continuing to evaluate that. At first, I think it did. And I'm now using various like deletion mutants and everything and trying to tease this out, not in cancer. And so I don't have a precise answer to that because that's my dissertation.
Okay, no worries. But then following up on that, there was a recent report that the interaction with Liprin can regulate the ability of LAR to oligomerize. Does Liprin have an impact on this? Can you see, is there any connection being established between these cancer pathways and some of the LAR binding proteins that might shed light on that aspect of things?
Yeah, I've actually looked at the PPF or Liprin a little bit in my studies, not in cancer yet, but I did read that paper and I got really excited over quarantine about it. So I'm continuing to examine that.
Okay, thank you.
Thank you.
I have a quick question. So you said that RF gets cleaved. I'm assuming that that is like the extracellular, intracellular, like it's cleaved there.
So at the membrane, they actually associate together, and when they are internalized, they do stay together because I've used, well, that's been shown previously, but also I've used like an extracellular antibody to stain, and I still see it in those vesicles.
So for your shRNA knockdown, it looked like you had differences in like the level of knockdown between whether you were looking at the kind of intracellular portion or extracellular portion. And so I was just curious if they had perhaps independent functions within this system. Does that make sense?
As in their extracellular versus intracellular roles?
Yeah, so like if your knockdown is preferentially, you know, kind of enriching for, I think like the knockdown of the extracellular domain looked less than like, let's say your full length. And so I was just kind of trying to like piece that together a bit as to like how those things are related.
That was something that we kind of struggled with, the knockdown. And so in my studies now, I'm using a CRISPR knockout, clean system. But we do think that it's not well understood exactly how like the cleavage affects it, or the processing. And so there's actually not like much known about RF in that way. And so I'm learning as I go.
And then just as a side note, I know you didn't touch on it, it's not necessarily your work, but as a potential target, Syndican, I know Syndican 1 a little more recently in pancreatic cancer has been a really interesting target and regulates pancreatic macropinocytosis. And so kind of looking at the regulation of that in pancreatic cancer through the PTPs would be a super interesting avenue, just as a FYI.
Yeah, thank you.
Yeah.
I've got one in the chat that was sent to me, and it says PTPRF is a part of the subfamily with PTPRD and PTPRS. Did you find any differences in the expression of those two proteins when you manipulate PTPRF? So like when you knock down or overexpress?
That's a great question. So as a control, I would also probe for D and S, like in my qPCRs. And overall, there wasn't significant changes. So I will say no, but it did vary a little bit, but I would control for that.
Hello, Ashley.
Hello.
Hi, I have 2 questions. So if you go to your immune fluorescence data with RFP tagged PTPRF, so how many % of PTPRF did you see are in the intracellular vesicles?
Let's see. I would say, I don't know, I don't want to put a % on it, but we do see both membrane and vesicle staining. So in single color control, we see on the membrane and then a few vesicles.
Yeah. So did you tag your RFP at the C terminus of RF?
Yes.
Okay. Just I've read a paper published in JBC in 2007, and the team, they identified another cleavage event happening to PTPRF between the transmembrane and the D1. So the cleavage leads to the release of an intracellular soluble fragment, which goes into the cytoplasma, and that specific species, they found it interact with beta-catenin.
Yes, I remember that paper.
Yeah. So do you think those intracellular vesicles, since your RFP is at the C terminus, do you think the vesicle one can be the cleaved RF species?
It's possible. I think in this current experiment, there's no way to tell.
Yeah.
Other than
so
using
if
like
you go to your
antibody
Western,
staining.
Yeah, so if you go to your Western blot data with the P subunit staining, so I assume your P subunit recognize the intracellular part of RF, right?
Yes, I have done staining with the intracellular P subunit
Yeah.
antibody.
Yeah, and I saw a band of lower molecular weight below your major band.
Yeah,
which
I think it's non-specific because that also shows up in my like CRISPR knockout cells as well. But
don't you think it's like
yeah,
decreased with your shRNA knockdown? Just like, because I can't see the Western blot
a little
clear.
bit.
Okay. Yeah, so I'm just suggesting you might want to look into the intracellular fragment.
Yeah, thank you for that
as
suggestion
as
and
it's,
paper reference.
Interact with beta-catenin directly, right? Also, I have another question. So in your introduction, you said PTPRF has both oncogenic and tumor suppressive function.
Yeah, depending
right?
on what cancer type.
Yes. So how do you think the tumor function of an RPTP is defined in different cell contexts? Because another example is in your tissue mRNA expression data in colon cancer. Another two RPTPs, especially PTPRD, which you said is a well-defined tumor suppressor gene. It has actually higher amplification rate compared to PTPRF.
Yeah, I noticed that. I think that it depends on signaling pathways. So it's tumor suppressive when it acts through the EGFR pathway. And so I think my study shows that RF in colon cancer acts through the Wnt pathway, and that's what makes it oncogenic. So.
All right, thank you.
Yeah, thank you so much for your suggestions.
Can I ask a question? So how do you think the tyrosine phosphatase LAR affects LRP6 phosphorylation?
So it's not direct because this is serine threonine phosphorylation, and RF is a tyrosine and phosphatase. There has been papers showing that LRP6 does have tyrosine phosphorylation, and I have tried really hard to show that it's a substrate, and I have not been successful, but not giving up. I think they do interact. There is some effect, but I don't know if it directly dephosphorylate it. So if you have any suggestions.
No, I mean, apparently it is indirect, so that's a possibility. I was thinking, so clathrin, you show an involvement of clathrin. I think Wnt signaling itself is very tightly regulated, or the Wnt localization is very tightly regulated, and that is also regulating its signaling function. So you might want to look into that. So where Wnt goes and how that is regulated. So that could be actually determining Wnt signaling strength output.
Yeah, I think it's like a co-regulator of Wnt. I think it's it helps just control like signaling levels, and so I'm kind of following up that line for my dissertation.
All right, cool, thanks.
Yeah, thank you.
Okay, I'm gonna go with we don't have any more.
Okay.
Okay, cool. Well, thank you, Ashley. And we'll email this out to you guys with the YouTube link to send to your friends, etc. And we have 2 people signed up for next month, so we'll be in contact soon. Jesse, you got anything?
Nope. Thanks for coming, and thanks for all the good discussion with Ashley.
Yeah, thank you.
Have a good weekend, everyone.
Bye.